Welcome back. Let's dissect this question. A researcher is performing a neuromuscular junction experiment wherein a specific immunoglobulin, IgG, is administered to cultures of innervated skeletal myocytes. Administration of antibodies directed against nicotinic acetylcholine receptors demonstrates a significant decrease in the number of receptors following administration of the IgG, and an attenuated response of the myocytes to electrical stimulation of the cells innervating the myocytes. Which of the following physiological mechanisms is likely to improve the response of these myocytes to electrical stimulation? So we see here there's a clinical vignette that involves some research, and the topic seems to be skeletal myocytes or skeletal muscle. That's very significant. Nicotinic acetylcholine receptors, we know, are found on skeletal myocytes. The response to electrical stimulation of the myocytes is a question whether or not it can be improved. So if we look at the choices A, B, C, D, we're looking for something that can improve the electrical response or the contractile response, because we know the principle of excitation, contraction, coupling. Electrical stimulation comes before contraction. So let's see if we can figure this out. Nicotinic acetylcholine receptors. Do I see anything in the choices that's going to give us an idea that nicotinic acetylcholine receptors are being stimulated? Well, acetylcholinesterase there certainly looks like acetylcholine receptor, at least the concept, and choline acetyltransferase. The other two don't seem to fit. So let's look at C first, inhibiting the activity of acetylcholinesterase. Well, we know if we inhibit the activity of acetylcholinesterase, here's the motor neuron, and here are the vesicles in the motor neuron that release acetylcholine. Here is the skeletal muscle, and the skeletal muscle has receptors called acetylcholine receptors, ACHRs. Part of the acetylcholine receptor is an enzyme known as acetylcholinesterase that breaks down the acetylcholine. If acetylcholine stimulates the acetylcholine receptors adequately, we'll get an action potential in the muscle and a contraction. So, inhibiting the activity of acetylcholinesterase should raise the level of acetylcholine in the synapse here at the neuromuscular junction and improve the response of these myocytes to electrical stimulation. Let's look at D, inhibiting the activity of choline acetyltransferase. Well, in the motor neuron, it's the motor neuron that makes the acetylcholine, and choline acetyltransferase, known as CHAT, is the enzyme in the motor neuron that we're looking for to make the acetylcholine. But if we inhibit that, we're going to inhibit the production of acetylcholine, the release of acetylcholine, and instead of improving the electrical response, we'll decrease the electrical response. Now, we have to be careful with all multiple choice questions that there aren't two possible answers that we have eliminated, because we're always looking for the best answer. Let's look at A and B. Phosphatidylcholine synthase doesn't seem appropriate because phosphatidylcholine synthase is the enzyme for making phospholipids in all membranes, nothing specific to nerves or muscles or excitable tissue, and would maintain the integrity of the membrane, but it certainly wouldn't be as good as acetylcholinesterase in improving the response to electrical stimulation. Now what about phospholipase C? Phospholipase C is part of a G protein coupled receptor mechanism for stimulating muscle, but not skeletal muscle. Smooth muscle and cardiac muscle respond to G protein coupled receptors. The receptors on skeletal myocytes are ligand-gated channels, and those ligand-gated channels are gated by or controlled by the acetylcholine. So B doesn't seem appropriate either. Therefore, we're going to go with C as being the best answer.